Dear viewers, this video is part 3 in the 4-part series on infections overview in Medicine for Anyone in the YouTube channel. The pathogen. It is important to break down pathogenesis, that is, evolving steps of a particular disease in humans, into different steps and stages. Most viral and bacterial pathogens enter the host via the mucosa of the respiratory, gastrointestinal, or genitourinary tracts, although some important pathogens are introduced by injection from insect vectors or through abraded or wounded skin. Most pathogens then must stick to a surface and have evolved structures that enable this. These are usually constructed from proteins, and many of them are complex and specialized. In bacteria, these protein molecules are known as adhesins and are often delivered at the end of long proteinaceous organelles called pili or fimbriae. The precise amino acid sequence of the adhesin, and hence its structure, can dictate which host and even which tissues within the host the bacterium sticks to, and can therefore play a major role in dictating host range, which hosts are the targets, and tissue tropism, specificity of the tissues it will affect. After initial loose adherence, enteropathogenic E. coli will stick more firmly to the intestinal surface via the non-fimbrial adhesin intimin. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Viruses. Viruses also rely on surface structures for host specificity, an example being influenza virus. Among several other mechanisms, the host range and the tissue tropism of influenza virus is dependent on the structure of its hemagglutinin molecule. On the respiratory epithelium, hemagglutinin binds to sialic acid, which is linked to galactose on the host cell surface. Human influenza viruses bind preferentially to a molecule which is abundant on human tracheal epithelium. Once established at a surface, pathogens have a wide array of possible strategies. They can stay at that surface and cause very little damage. Bacteria such as Haemophilus influenzae and Neisseria meningitidis are good examples of this. Only as a result of some unknown and rare set of circumstances will these bacteria move into the bloodstream to cause septicemia and sometimes meningitis. To survive and spread in the blood, bacteria have evolved a range of molecular strategies to inhibit the activation and activity of complement and to avoid or resist phagocytosis. Alternatively, the bacteria can stay at the mucosal surface and cause considerable damage by direct invasion and destruction of the tissue, by inducing a damaging inflammatory response, or by elaborating a toxin. Enterotoxigenic E. coli makes labile toxins and stable toxins, which will usually result in watery diarrhea, whereas enterohemorrhagic E. coli can make Shiga toxin, which is spread systemically and acts at a distance from the gut, with severe consequences such as thrombocytopenia, that is, lowered platelet numbers, and kidney damage, leading to hemolytic uremic syndrome. Note: hemolytic uremic syndrome (HUS) is a condition that can occur when the small blood vessels in your kidneys become damaged and be inflamed. This damage can cause clots to form in the vessels. The clots clog the filtering system in the kidneys and lead to kidney failure, which could be life-threatening. Similarly, viruses bind to specific host cell receptors, and the distribution of these receptors can determine the range of pathologies associated with the infection. virulence factors for us humans, actually fitness factors for the microbes. Almost any gene product that has been identified as being required for infection has been called a virulence factor. For bacteria, many of the genes required for host exploitation might be better considered as fitness factors, but are no less important in the consideration of infectious diseases and how to combat them. Those that are include adjacents and toxins, and also various metabolic pathways that enable the bacterium to survive for long enough and grow in the host to cause damage. Viruses, on the other hand, are obligate host parasites. They tend, with notable exceptions such as herpes viruses and pox viruses with genomes of 100 to 200 kb, to have rather small genomes with few genes. Note, 1 kilobyte is 100,000 bases. Bases. See general information on genetics, the gene control of cell function in Medicine for Anyone in the YouTube channel. The bacterial genome varies in size from several hundred thousand to over 10 million base pairs. The viral genome generally bears less than 1 million, with the smallest known RNA virus genome being only 1700 base pairs. Therefore, in most viruses, each gene is required for exploitation of the host in some way and is highly likely to be a fitness factor for the virus. It may well be appropriate to consider them as virulence factors for us, the humans, in pathogenesis. In considering virulence versus fitness in evolution, we might ask, why do pathogens cause damage rather than simply existing in harmony with their host? There are many possible answers. One possible answer might be that by inducing a certain pathology, the pathogen liberates more nutrients for itself and or facilitates its transmission to another host, preferably in most cases before it kills its original host. These microbes have greatly and confidently assured their places in their chosen host, we humans, and are so confident that they have decreased their gene repertoire to maximize their success of replication in lesser time. Kindly note that small genome size is perfectly suited to replication, in which each infected host cell produces many copies of the microbe's genes from a single template. Such exponential replication places a premium on small genome size. The smaller the genome, the faster it can replicate. Adaptation to the environment. It is now clear that bacterial pathogens sense the environment in which they live and alter gene expression profiles accordingly to enable exploitation of and survival in that environment. That means the microbes are great adapters to new, changing, and hostile environments, our bodies, and are one step ahead of us as far as survival and preservation of their progeny are concerned. For example, a food poisoning bacterium such as Salmonella enterica might be living on a nutrient rich piece of meat, but at a cold temperature. The meat might then be cooked, providing the bacterium with heat stress. If the meat is undercooked, the Salmonella will survive the heat stress by expression of different heat shock operons, which incidentally might also lead to the expression of genes required to survive subsequent assaults in the host. Note: operons. An operon is a cluster of genes that are transcribed together to give a single messenger RNA mRNA molecule, which therefore encodes multiple proteins. Such an mRNA is typically found in prokaryotes, example bacteria. On entry into the mouth, defenses such as lysozyme and IgA must be overcome, and on entry into the stomach, a very low pH is encountered. Gene expression will again change in the Salmonella such that genes for acid tolerance and acid resistance are now to the fore. Once the bacteria exit the stomach, the pH will change again and they will be assaulted by bile salts and many other defense mechanisms until they arrive at their point of attachment to the small intestine. These carry entirely different and adverse circumstances, call for urgent and appropriate gene expression for a bacterium to establish itself in a host animal. A membrane protein in the microbe senses the external environmental cues, and the second is an intracellular protein that binds to DNA and either activates or represses the transcription and expression of sets of genes usually called regulons. A signal is transmitted from the sensor to the activator repressor when the environment changes. That is, the signal passes through the cell wall and enters the cell wall. Signal transduction is achieved via histidine protein kinases. The signal perceived is relayed to the nucleus DNA. These two component systems are very common in bacteria. A crude analogy would be the territorial army sends special messengers in these days of internet by dedicated email or WhatsApp to the army headquarters, which dispatches appropriate signals to different units to produce different types of forces, infantry, navy, and air combat units, and logistic support for the invasion against the enemy, us humans, of course. Those bacteria that can live in numerous environments tend to have many more of them, example 90 in Pseudomonas, whereas those bacteria that have become adapted to a lifestyle in a particular host have very few, example 2 in Chlamydia, 1 in Mycoplasma, and there is often a concomitant loss of genomic size. This is becoming ever more important imperative, with major concern being expressed regarding the rapidly increasing prevalence of bacteria that have evolved to become resistant to the current set of antimicrobial drugs. A further consequence of resistance to antimicrobials is that many of our current therapies for a range of diseases such as treatment for cancer and major joint surgery require extensive coverage by antimicrobial drugs so that secondary bacterial infections do not occur. If resistance continues to increase in prevalence, these therapies will also be severely compromised. Dear viewers, this is the end of video part 3 in the 4-part series on infections overview in the Medicine for Anyone in the YouTube channel. Thank you for watching.